Some of you may not know Clint. He just graduated from fellowship and joined Northwest Asthma and Allergy. This topic, I made a slightly different topic here. I called it the elegant something or other, major histocompatibility and TCR receptor. And this came to me because I've been studying with the fellows to prepare for the boards. And when we went on this topic, it was either something I never knew, didn't exist when I was a fellow, or I forgot all of it. But anyway, it struck me that the immune system is amazingly elegant and intricate, probably too much so, because if you make it this elegant and intricate, they make room for mistakes. So unless they were thinking about the careers of future immunologists when whoever created this system. But I knew Clint would do a good job with this topic. And so we're going to let him go ahead.
We're going to set the bar a little bit lower than do a good job. Let's try to shoot for adequate. So I'm Clint. I'm one of the recent graduates. So we're going to try to go into whether you want to call it the intricate relationship or the elegant relationship between major histocompatibility complexes and the T-cell receptor. I will try my best to talk about it in under an hour. I have no disclosures. And then our objectives today are to understand MHC, what it is, the different types, non-traditional MHCs, which I know is kind of more newer developments, kind of what we understand. We want to understand how cells do antigen presentation and then how T cells develop. I won't spend a ton of time in cell signaling. That would be a lecture in and of itself. I won't spend a ton of time on B cells or antibody production. I will try to cover some topics that are more pertinent to the clinicians. But initially, we're going to kind of have to go a little bit on a journey into the weeds, into some basic science topics. And I will try my best to make them a little bit less complex. So I will try to keep things flowing. If there's any questions along the way, just unmute, we can talk. This will be a little bit more informal. So for background, just so that everyone understands, depending on when you trained, when you did, took your boards and everything like that, MHC, HLA, TCR may not have actually been understood at the time. So initially, all that was really known about antigen presentation or T cells in general was in the setting of transplanted organs. And so we knew that people that had a non-identical match, they had failure. And we never knew exactly why at first, but we would always try to have cross-matching as much as we could, but it wasn't on the molecular basis that we have right now. And then in 1937, the guy that you see in the top right picture, his name is Dr. Snell. Him and Dr. Gorer identified a specific gene locus, specifically in mice, that prevented engraftment whenever they were inbred, but they had just one specific gene difference on their chromosome 15. And they named it histio, meaning tissue, compatibility, meaning that they were kind of aligned whenever they enabled engraftment. And it was similar to the antigen 2 blood group. So they called it histocompatibility 2. And that kind of formed the groundwork for all of MHC. MHC is kind of a big term. So it's kind of like animals, and then we have mammals and everything else underneath it. But initially, it was really only known in the setting of mice. We hadn't specifically identified it in humans, and we also didn't really know about it in the setting of anything other than transplantation. And then in 1974, Dr. Zinkernagel and Doherty, they identified more than just the histocompatibility 2 to find in T cells, specifically CD8 cells, that in vitro mice that were infected with LCMV virus were able to target and lyse infected cells, but whenever it was matched between antigen presentation and specific T cells. But they were always restricted by the cell type. And so this kind of helped guide us into the next phase of our understanding of MHC and TCR, where it did more than just block transplantation or affect transplantation. And
Clint,
then both,
in 1974, did they actually know that there were CD8 cells?
they did not know that they were CD8. So they had identified that there was a marker on it, and eventually it became CD8. But they knew that it was a restricted cell type to fight in intracellular viral infections. And subsequently was found to be CD8. And then in 1996, so for reference, in 1980, Snell and Gore received the Nobel Prize. And in 1996, Zinkernagel and Doherty received the Nobel Peace Prize. And within a month of them receiving their Nobel Prize, we actually had the first identified X-ray crystallography picture of the MHC peptide TCR complex, which is extrapolated in the picture on the right. But so this is not the exact same one that they had, but it showed the current antigen presentation model that we think about for all infections, not just LCMV virus in mice. And then this was actually in humans is where we identified it. And so what we're going to kind of focus on is when things go right and when things go wrong. So first we have to understand when things go right, kind of what happens. So we have to make the major histocompatibility complex. We have to load the major histocompatibility complex. We also, on the other side of things, while the MHC is developing, we have to make a T cell that makes a TCR. It has to be functional, it has to signal, and then they have to interact together. So there's many steps along the journey to try to make sure that things get right. So focusing specifically on MHC, sometimes you'll hear us talk about it as the major histocompatibility complex or the human leukocyte antigen. So MHC is kind of for all animals, HLA is specific to humans. So HLA or MHC is one of the most interesting kind of sections of our genome. If you actually look at it, it's the most diverse location as far as germline location, entirely because they have to be diverse in the way that they can present antigens. And there have been identified over 250,000 different alleles for HLA as of January 2020, which was the last time that it was published. There's a database that kind of tracks it. And it was tracked to chromosome 6. And so in this region, it is always inherited in blocks. So this region is not split, it's not cut. You always receive one block from mom, one block from dad. And different than if we use the term MHC1, MHC2, this gene locus has specific class restrictions that is in the actual genome, where they have a lot of really important genes that are kind of stacked in there. So they have the loading genes. So, and we'll talk about these. So Tapacin, TAP, HLA-DM. They have both the MHC2 structures. So those are DM, DP, DQ, DR. And then they have the MHC1 loci, which are HLA A, B, and C. They have the non-traditional HLAs. And then they also have a lot of our complements, a lot of our signaling cytokines like TNF, lymphotoxin. And so what happens whenever you actually, so if you look at the bottom right, whenever you actually have a mom and a dad, mom has her own sets, dad has his own sets. You kind of inherit them in block. And so all the children's will either have, 50% from mom, 50% from dad, and they're expressed as a composite of both parents. So you always express all of dad's that you receive and all of mom's that you receive. And so, which is one of the reasons why whenever, a baby or anybody needs a bone marrow transplant, if you have a sibling, you have a 25% chance of having a perfect match just because of the genes that are specifically identified. So in this instance, in the bottom right, bottom left, if dad has all of the, A1, B, all of the red ones that are A1, B8, DR17, mom has A24, B7, DR8 is that, and then the blue options, you have a 50-50 shot for which the specific ones of dad to be received, and then you have a 50-50 shot for the ones that mom's going to give you. It's a composite, it gives you a 25%. So going into each MHC specific, so this is not talking about the genes, this is actually talking about the class 1 MHC or HLAs. So class 1 are traditionally on all nucleated cells. So they are constitutively expressed, both showing self-antigen, but also presenting any intracellular stress or protein. They can be upregulated in the setting of a viral infection, which we signal through interferon alpha and beta. They are a lot of our innate pathways that signal for stress, including PAMPs and DAMPs, can increase class 1 expression. So like your TLRs, your nod-like receptors, your RIG-like, all of those can specifically upregulate your MHC class 1 by also increasing your interferon alpha and beta, which are used for viral infections. The interesting thing is that our antigen presentation cells, or dendritic cells, our tissue macrophages, they can allow cross-presentation. So they take in damaged cells, they phagocytose them, they break them down, and then they represent them so that CD8 cells can be activated. On the right, you can see the structure. So it does have, we always number from the furthest distally and kind of coming back. So it's alpha 1, alpha 2, alpha 3, closest to the cell membrane, and then you have the beta 2 microglobulin that is non-ionically linked. They have an immunoglobulin kind of loop, which you'll see with disulfide bonds, which are those little red dots right there. And then they present right in the cleft the peptide. The peptide groove for all MHCs are the most variable or the most polymorphic location. So most of the HLA polymorphisms or different alleles that you'll see are actually in a specific antigen binding groove. MHC class 1 are named, as far as the nomenclature, we have HLA A, B, and C, which are your classic MHC class 1. Because we've now have gotten to the molecular stage, we actually have further broken down the nomenclature. So instead of saying HLA A, B, and C, we actually break it down in specific breaks. And so you'll hear it said, like an example that I wrote there is 01, 100, 01, 01. And so that goes kind of step by step of, it's the allele group, it's the specific protein, it's the DNA substitution, and then it's the non-coding region, just to make things more complex. So whenever you talk to anybody, you can sound really smart when you start throwing out all of those numbers. MHC1, because they present intracellularly, and we'll talk about how they load the MHC, or how our cells load MHC in a second. If you think about it, the way I kind of always conceptualize it, you can think about it like a baseball glove or like a hot dog. Whenever you load the hot dog bun for MHC class 1, the ends are cut off so it fits specifically into the groove. It's a little bit of a smaller protein, so they only present 8 to 11 amino acid residues in the binding cleft, and the binding cleft is between alpha 1 and alpha 2 loops. It is restricted to MHC1 only interacts with CD8. So if you think about it like a lock and a key, or like a handshake. So if you reach out for your hand, that's your MHC reaching out. Someone else reaches out their hand, that's the TCR. And then if you want to do a double handshake, that's like the CD8 coming in to bind and make sure it's a really great handshake so you can celebrate it. Even though I know right now we're not, and no one's doing handshakes because that's like a faux pas, but that's kind of how you can conceptualize the CD8 is the extra hand coming in to kind of lock the handshake tightly together. Whenever we load our MHC class 1, it is an incredibly complex process. So it starts with one of two aspects. So either we take in damaged or dying cells, we break them down in the phagolysosome, or whenever a cell gets into when a viral infection gets into our cells, remember these are intracellular pathogens. We make the viral protein, we make the mRNA, our body recognizes it as foreign, we put a coating on it through a lot of our intracellular pathways to then signal to the proteasome to break it down, and then it takes the broken down version and it uses the TAP protein, which is seen right here. I don't know if you can see it. That's what passes the broken down peptide into the endoplasmic reticulum that is waiting to combine with the MHC complex. The MHC comes from the nucleus, its own self, comes into the endoplasmic reticulum and is transcribed via mRNA to make a protein. That protein is then has two separate sites. So they have the alpha side and then the beta 2 microglobulin. It's initially empty, and then through the interactions with some chaperone proteins like tepacin and calreticulin, it then loads the specific signal or the specific peptide into the binding cleft, and then progresses out to show it to the surface to then interact with cells. So it's a multi-step process that is kind of built in complexity to make sure that we only present the right cells, the right signals or infections. And it has many potential kind of stopping situations to prevent self-antigen from always being expressed and then causing autoreactivity. Any of these peptides along the way can cause an issue or a not efficiently produced MHC molecule. And so we got smart since it was the MHC1 molecule that we named the syndrome that happens as the Bare lymphocyte syndrome type 1. So I apologize for the typo there. It should say Bare lymphocyte syndrome type 1. Because they do not make MHC1, whenever our CD8 cells are developing, they never get the signal from MHC1, so they never get a survival signal, so they are not made. And so this could be due to not making the beta-2 microglobulin. This could be due to not making the TAP protein or the chaperone protein inside the endoplasmic reticulum, which is tepacin. And interestingly, even though these are usually intracellular pathogens that kind of stimulate the MHC1 molecule, is you can see a spectrum of disease. And so with Bare lymphocyte type 1 syndrome, it depends on which step along the way is having problems. And so some people, we don't even identify until adulthood that they just have primarily bacterial infections, and usually it progresses on to bronchiectasis. And it's usually due to Staph aureus, Strep pneumo, Pseudomonas, and Klebsiella. And they can have, if you actually have further upstream, so either the transcription factors, so identified that actually make the proteins, then you can actually have a more severe phenotype, which also has fungal infections. But the vast majority just may have bronchiectasis and granulomas and actually hypergammaglobulinemia to compensate. And for the rest of us clinicians out here, the mRNA vaccines, this is actually one of the ways that the mRNA vaccine, so not only do we make antibodies to it, but whenever we have our antigen presentation cells see mRNA, they transcribe it into a protein, they break it down, and you actually have a CD8 clonality that responds to the specific vaccine. And so this is one of the other ways that we typically have an immune response. So whenever we are all talking about, waning titers, whenever some of the immunology individuals out there are saying, titers are not always telling the full stories, we do still have memory CD8 cells that are there to fight infection, which is probably more efficient to actually fight damaged cells. Len, did you have a question?
Yeah. How does our system know which 8 to 11 amino acid peptide is the relevant one to make an immune response to, from the whole thing it chops up in the proteasome? Who's inside there deciding, oh, this is the important part to recognize?
Magic. So some of it's done randomly. Some of it it's chopped up randomly. Some of it, we have a consistent. So if you look at all people, they have the same sections that are actually chopped up and segmented off. So there has to be some level of specificity that we haven't fully identified. Some of it is related to the specific area that can be ubiquitinated. So you have to have an amino acid that can be charged or remove electrons to then add on ubiquitin to it. And so that probably is one of the main reasons. But whenever it actually chops it up, there is some level of kind of flexibility or kind of spontaneity, so that it can actually enable different versions of it. Because if you think about it, the peptides that they make, we could have some hidden areas, you can have some intracellular areas, and so you have to be able to mount an immune response to something that could be seen externally as well.
So do we make some T cells that are inefficient, that don't recognize the important protein, and just by luck we make enough of them that we also make an effective CD8 cell?
So remember, we're talking about the MHC side. So this is the antigen presentation side. Yes, we do. And so that's where it becomes a survival of the fittest, which we will talk about downstream in a second. But T cells, so it's the same concept behind B cells. Whenever we have somatic hypermutation, where you actually make a more efficient receptor, a more efficient antibody that then causes the signal to propagate and grow in a specific clone to kind of continue to grow. It's the same concept behind here, where the T cells that are most efficient at recognizing a specific antigen that actually functions and that we actually will see kind of survive. Because if you don't continue to get that signal, just like, you know, I myself, we get lazy, we kind of fall back into apathy or anergy, which can happen. So transitioning from MHC class 1 to MHC class 2, MHC class 1 primarily are more for helping kind of mount a separate arm of our immune system. So these ones actually require antigen presentation cells, so not all cells make them. B lymphocytes typically constitutively express MHC, which kind of goes into the model of B cells and T cells needing to interact constantly, whereas your tissue macrophages in your dendritic cells, they actually upregulate or they have an inducible MHC class 2. It's typically stimulated by interferon gamma, but it doesn't always shunt it towards a Th1 phenotype, and we'll talk about what I mean by that. This is traditionally responding to extracellular peptides, and then the MHC class 2 kind of helps push CD4 positive, which is the restriction for MHC class 2, towards a helper function. And I apologize, there's also a typo on this at the bottom of the slide. So MHC class 1 for the nomenclature does have, it's identified as DPDQDR. It has an alpha and a beta chain, and similarly, we number it from further out to further in. So it's alpha 1, beta 1, alpha 2, beta 2. They are non-ionically linked, and they are similar to MHC class 1. They do have an immunoglobulin-like structure, just showing that they have that beta chain or that beta strand alpha loop kind of over on the right, and then they actually have the binding cleft. The binding cleft is actually alpha 1 beta 1, so I apologize. Oh no, I'm sorry, that is correct. I misread. So this one, if you're thinking about it again in terms of like the hot dog bun and hot dog concept, it allows like a foot long. It allows a longer amino acid, mainly because then it has to be able to respond to a little bit more of a broader infection, entirely because it only has a limited amount of cells that can present. So where all cells present MHC1, MHC2, since it can only be done by antigen presentation cells, it allows itself to see a little bit of a broader scope. Once again, it only sees amino acids, so it doesn't see other non-amino acid structures. And then the CD4 marker, similar to the handshake, it's the second hand that kind of comes in and tightens on it, and then it actually binds past the binding cleft and down towards where the alpha 2 beta 2 typically can. So if you see right here, this is actually where the CD4 kind of sinks in, and it's like a key that kind of holds it together. And once again, the binding site actually has the most polymorphisms. So we talked about loading MHC class 1, so we'll talk about loading MHC class 2. So remember, they are extracellular proteins. So we take it from the extracellular space, we bring it into our cells via phagocytosis or endocytosis. We chop it up via lysosome, the lysosome coming in and fusing to make the phagolysosome. And then at the same time, our antigen presentation cells are making the structure that becomes the alpha chain, the beta chain, and they put that into the endoplasmic reticulum. It binds to, it's identified as this pink little block here. It's called the invariant chain. The invariant chain stabilizes the alpha and the beta strand of the MHC complex to sit together, so they actually can hold it tightly together. And then one of our non-traditional HLAs, which is called HLA-DM, similar to Tepacin, which stabilized MHC class 1, and HLA-DM comes in and stabilizes the MHC2 complex and cuts the invariant chain to remove it and make what's called clip. And then that enables the loading of the antigen into the MHC2 complex, which then pushes it to the cell surface. HLA-DM does not interact with any of the antigens, so it's not considered a classic MHC molecule. So it is, even though it's in the same gene loci, even though it has a very similar interaction with the MHC2 complex, it is not considered an MHC class 2 molecule. And then APS, similar to Bayer lymphocyte type 1, Bayer lymphocyte type 2, named due to issues producing the MHC class 2 molecule, can happen with anything along this way. So if you have an issue with HLA-DM, if you have an issue with either the alpha or the beta 1 or the transcription factors that signal to it, which is called the MHC2 transcription activator, which is also called CIITA or C2TA. If you do not make an MHC2 complex, you will not make CD4 cells. So this is another form of a severe combined immunodeficiency. They have invasive viral and fungal pathogens similar to SCID. And then whenever we all get our flow cytometry, especially if we do it either from the lab at UW or over at Seattle Children's, whenever you see it says HLA-DM, I'm sorry, HLA-DQ, you actually see that tells you if they actually can have bare lymphocytes in them. So you're actually getting that information whenever we all check flow. We may not realize it, but that's one of the things that they typically do, and that's looking on B cells which constitutively express MHC class 2. So one of the newer things that's kind of been out there, and we think that it probably plays a role in kind of the development of allergy, are the non-traditional MHC. These present non-peptide antigens. They are MHC-like in that they present antigen, but they don't have the same kind of restriction as MHC1 or MHC2, which can only do amino acids. So these can put out lipids, polysaccharides, some small molecule haptens, including metals. And so this is one of the things that we think may play a role with some of like the allergic contact dermatitis, the initial development of, or playing a role in food allergy and atopic dermatitis. It signals through NK T cells, which are kind of more of a hybrid between the adaptive and the innate immune system, as well as the gamma delta T cells. They don't really process the antigen, so they don't really chop it up, and there's not as much polymorphism, so there's not as many changes in the germline genetics, but it kind of can play a role as far as the development of diseases. And then two of the other ones that I at least was asked about on my boards, and so I think that it's important to know, are HLA-G and E. HLA-G is important in the development of fetal tissue. So it's actually an interesting HLA where it interacts with NK cells and increases the production of regulatory T cells, and it down-regulates the expression of paternal MHC on the developing trophoblast and the developing placenta to actually prevent cross-presentation of maternal MHC, which would then signal to mom's tissue that there's something foreign or a parasite growing in her uterus, and so then it would attack it. So it's one of the ways that we actually protect the baby to actually have a functioning pregnancy. So the kind of using alternatives to protect the developing fetal tissue. And then HLA-E is an inhibitory signal. So NK cells kind of go around looking for missing self. So if a viral infection wants to try to escape the immune system, they can try to decrease how much MHC class 1 they make. And MHC class 1 and HLA-E can actually, the presence of it can downregulate NK cells. And so just like MHC1 activates CD8 cells, the absence of it, or the absence of HLA-E, which coincides with the production of MHC class 1, signals for NK cells to attack that cell. And so the absence is associated with NK activity. It's never been associated with some of like the overactive NK cell opathies, so like your HLH, but it is a potential risk factor. So we're going to talk for a second. I know everyone here has already probably seen the diagram on the right many times, kind of the progression for T cell development. So I'm going to I've been mainly focusing on the alpha beta T cells, and I'm going to mainly kind of continue to focus on that, mainly because less is known about gamma delta T cells. They're not as specific. They are they're very efficient, but they're very important, but we don't fully understand them as well as we do the traditional T cells, which are alpha beta as a T cell receptor. So starting from pluripotent hematopoietic stem cells, we progress along to the common lymphoid progenitors, which then signal to either become B cells or T cells based on the transcription factors in the milieu that they're in. Then the T cell side either becomes innate lymphoid NK T cells, or it progresses down to become true T cells, which then have to go to the thymus. So this is kind of, it requires the bone marrow to make the lymphoid progenitors and the T cell progenitors, and then they have to leave the they have to leave the bone marrow and go to the thymus, where they have to generate a functional T-cell receptor to kind of mature and develop, which typically happens in the thymus. But then they leave the thymus as still relatively naive because they don't actually have an effector function, and they get activated in the periphery in the lymph nodes to then kind of go on and have their full impact. And so when a T cell enters the thymus, I know all of us remember this and we cringe at this. We have our baseline germline DNA, and then there's signals in the thymus to then chop down our variable, our diversity, and our joining segments of our DNA to make a new rearranged DNA that has to be functional and produce a T cell that does not have an inappropriate stop, that has kind of a perfect amino acid sequence that can interact with your MHC and then transcribe it. And so the way that it does it, it takes an unarranged locus, which you see kind of here in the middle. It kind of brings two areas of the genome together. It cuts out that cleavage site and keeps them next to each other. They add amino acids to give diversity, and then it makes a specific functional genetic strip that then can be transcribed and become functional mRNA to make the T cell. The cut out version is what we call TREX, or T cell receptor excision circles, that we produce. You have to have a functioning thymus because this takes place in the thymus to make our T cell receptor. And so that's one of the things that we look for in severe combined immunodeficiency, or that we're screening in all 50 states at this point, and is recommended to screen all countries worldwide as well. And it's one of the first sites that we can tell if either there is not a thymus or there's not T cells that progress to the thymus and kind of undergo this change. So on the left side of the screen, so T cells come into the thymic cortex. They're what's called double negative T cells, which means that they do not have CD4 on their surface, they do not have CD8 on their surface, they do not have a T cell on their surface. Then they kind of progress in the cortex, and they have to make both CD4 and CD8 because they don't know which direction they're going to go, and they have to make a functional T cell receptor. And then they have to undergo selection. So positive selection kind of reinforces that they are doing the right thing, that it's that they have a T cell that interacts with MHC. If there is no recognition, they die by neglect. It's kind of like the Goldilocks principle. So it has to be just enough, but not too much. And so initially it has to be just enough, right? So it has to make it and has to interact, has to be functional. And then after that, once it kind of progresses in the thymus to the medulla, it has to be specific. So then they go from double negative when they enter to double positive in the middle to single positive. So either CD4 or CD8 based on which MHC they interact with the best. So either MHC1 or MHC2. And so then it becomes specific to either CD4 positive helper T cell or a CD8, which we call cytotoxic T lymphocytes. If they bind too strongly, then that's the sign that they are autoreactive, that they will attack self, because we're only exposing them to self-antigen in the thymic medulla. And so if it's too strong, they get the signal for negative selection to actually die by apoptosis entirely, because we do not want autoimmunity to develop. This is also one of the theories and it's been shown with a specific issue in the medulla with antigen presentation cells in the medulla, where if they don't kill off these T cells and that are too hyperreactive or autoreactive to self, then we can actually develop down into autoimmunity downstream. Some of the cells that bind somewhat tightly to our own self antigen and MHC then do progress to become T regulatory cells, which you see at the bottom. Those are actually incredibly important for maintaining peripheral tolerance to prevent our cells from becoming too autoreactive and kind of shutting down the immune reaction that we see with any infection. And so if you either do not make the precursor lymphocytes, you do not have the thymus, then you will have a combined immunodeficiency that we can see at birth. And so you can have a positive TREX screen for any of the steps as far as cutting out the genome in here, for if you don't make MHC so that it can't interact over here with CD4, CD8 cells, or if you don't make a thymus, so like in the classic DeGeorge, which is due to a 22q deletion syndrome, or 10p genetic deletion or charge. So we talked about making the MHC, we talked about making the T cell. Now we have to kind of have the Lego pieces come together and interact with the right protein kind of sitting in there. And so this is the crux of probably of our specialty and the kind of the most delicate interaction, entirely because unless our antigen presentation cells recognize the antigen as foreign, they won't make a second signal to then signal to the T cell. So this actually is all driven by our antigen presentation cells. They have to be smart enough to recognize that this is foreign, this is a bad guy, and that we need extra help to the party. So this is kind of where innate meets adaptive. And so in the periphery, the T cells leave the thymus, they kind of travel around in the bloodstream, they come out into the lymphatic tissue, and in the peripheral lymphatic tissue, they look for antigen presentation cells that are showing markers of infection, stress, anything like that, to activate and mature. And there's also another checks and balance, because if we do not have the second signal, then the T cells won't proliferate and grow into a clonal expansion. If the T cells don't find a match, they go back into go back onto dating websites. They go back through the lymphatics back into the peripheral blood and try to kind of circulate around again to find their perfect match. It requires a lot of extra signals, and we'll talk about that in a second. And this is one of the redundancies that's built into our immune system to prevent inappropriate activation. We have to have the second signal to then cause T cell activation differentiation. And the way that we get different T cells is based on our antigen presentation cells making a certain cytokine environment, and then the generation of a synapse. So they actually have to come together. So for the cellular synapse, it varies on how long it takes. So a lot of what we've been talking about is kind of thinking in isolation of one MHC sits against one T cell, and then the second signals come along. Technically, it takes around 25 to 30 T cell receptors and MHC to come together and interact for there actually to be true stimulation. And so what initially happens is somewhere around 25 MHC TCRs come together. It signals through the second signals if they're present. So those things are kind of, if you see along the right side up here, is all of these markers have to come together. So the CD4, CD8 has to sync onto the MHC class 1 or MHC class 2. Then we have to upregulate some of the markers that kind of signal to propagate an immune signal, such as CD28 binding to CD80 and 86. And then the cytokine milieu or the complement system kind of activates further signals to kind of progress, and it kind of stays isolated in the core where most of the interaction goes on. But then as we get further out on the actual kind of the target for whenever T cell sits up against a cell, you have extra signals that can only help propagate it. And then you have to rearrange the cytoskeleton, which is where you see a lot of your combined immunodeficiencies. So like with Scott Aldrich, where they don't generate that cytokine restructuring to then pull the T lymphocyte directly up against the either antigen presentation or the infected cell to then cause signaling. And so CD4 cells are a little bit slower because we want them to be a little bit more accurate. So we want them to make sure that they are attacking the right organism. So initial activation of CD4 cells requires 20 to 24 hours to fully activate, where it sits directly on the surface of that same cell. And if we don't generate the cytokine restructuring, the cytoskeleton restructuring, if we do not have a strong enough connection, either because of the T cell and the MHC interaction, or we don't have the second signals, then the T cells will disconnect and kind of go back in the periphery. Because CD8 cells have to respond a little bit quicker, and they actually have to kill damaged or dying infected cells, they require less time for initial activation, which is only 2 hours. Subsequent activation is much faster, both for CD4 cells and for CD8 cells, but initial activation does take a little bit longer. And so whenever we differentiate our cells, so I know that we all are used to talking about it in terms of Th1, Th2, and Th17. So we're going to start on helper T cells for the right, so that right figure. It is immensely more complicated, and we are finding different subsets that have kind of different function. So your Th1s are your kind of your classic cellular immunity or technically bacterial immunity that kind of progress to help with neutrophilic clearing. Th2 initially is for helminth infections or parasitic infections that technically will clear up. It has also been shown to be due to atopy or allergy. And then a lot of your other regulatory cells use the same stat sequences, use the same kind of initial naive T cell structure, but then based on the cytokine milieu, whether that's pro-inflammatory, so IL-12, IL-6 versus IL-4 versus TGF-beta, kind of will progress in a direction that is preferential for the job that our immune system is going to. And then some of them, the follicular helper cells, are then to stabilize the lymph node structure. And then just to make everything more complicated is once we actually make these T cells, they inhibit or activate themselves and other versions of it. So Th2 cells do not like Th1 cells, which do not like Th2. And so they try to downregulate each other and interact in that way. Whenever it comes to CD8 cells, it's a little bit more straightforward. So the left side, kind of the lower marker is typically how the T cells kind of progress from naive to activated to memory to effector memory to kind of terminal effector, and then they can actually lose that activation. CD8 is slightly different. I realize this is actually a picture for helper T cells, but CD8 you can actually have a version. It's based off of their CD45 progression, and you can actually go from RA, which is immature, to RO, which is memory, and then you can actually have, for CD8 cells, you can make, just to make things more complicated, a version that reverts back to an immature, and it's called TEMRA, or T cell effective memory, RA, which is still an immature marker, but it is efficient at kind of turning back the clock, and so that it can actually continue to activate and affect a cellular cytolytic effect. So we got into the weeds. That was a lot. And so we're going to try to go to how we can actually apply all of these basic science principles and how they can actually help us. So when things go wrong, there is a lot of information that's coming out there as far as how antigen presentations or polymorphisms, including methylation of the DNA to kind of suppress some specific alleles, can lead to disease processes. So specifically an allergy, for asthma, food allergy, atopic dermatitis, for antigen presentations in the lungs and in the lymph nodes. There is a lot of information for drug hypersensitivities, but there's only a few that actually are kind of shown that we need to screen for, we need to worry about. And then it makes sense that different changes to the way that we present antigen from our antigen presentation cells to our T cells. It would also have issues with overactive autoimmunity, kind of autoreactivity, as well as susceptibility. So I am going to go through some of the specific HLA markers that are correlated with disease. The traditional thought is, unless you're using this all the time, you're probably not going to remember it. And so these are only really useful if you see this all the time, or if not, we can all just look them up. And there's a growing body of literature for each of these. So things that probably matter the most to us is drug allergies. So whenever a drug comes in, most drugs are either prodrugs or are small molecules that can't technically on their own activate the immune system. And so we don't fully understand why in some of your severe cutaneous adverse reactions to medications, we have CD4, CD8 activation specific to that drug. And some of it we've kind of shown is either related to reactivation of herpes virus families, so HHV 6, HHV 8, as well as some of the structure of the molecule. So they've come up with different kind of theories on why this happens, as far as the hapten-prohapten model, where whenever a hapten interacts here in the middle, or here in the left first, is that it interacts with a protein that we all make, so things like albumin, that then enables the small molecule to sit in the middle, which then can kind of go right into the groove between your for your alpha beta T cells, right between the either the alpha 1, alpha 2 for CD8 cells, or the alpha-1 beta-1 structure. It could also be that the alpha-1 beta-1 is activated to the specific protein that sits at the end. I'm sorry, the specific hapten that sits at the end of the protein, which is the pharmacointeractive model. And then there's the more recent thing that we've kind of thought about is the altered peptide repertoire model, so that the small molecule hapten sitting with the protein pushes the protein up into a position that activates the T cells in an unusual way that we wouldn't typically do. And that's why you can have first drug reaction or first drug exposure reactions instead of what's kind of thought of as, you know, like a delayed hypersensitivity where it takes time to kind of develop, where you can actually have it with the first drug. The classic and the only one that really kind of bears the most mentioning is abacavir, used in HIV treatment. It has the most body of literature. It's one of the few ones that is actually recommended to screen for before you start therapy with, and it's related to a specific HLA-B star 5701, I believe. And so what happens in SJS-TEN or in DRESS is that with different drugs, and there you kind of see them along the right, in specific patient populations, they have higher incidence rates of specific HLA types. And those types, whenever they interact with the medicines through one of the models that we kind of talk about, either the pharmacointeractive, the altered peptide, or the hapten-prohapten, activates T cells that cause the severe cutaneous adverse reactions. While some of these do have an increased risk, it is not yet recommended for most of them to screen for these prior to initiation of the drug, with the exception of abacavir and likely carbamazepine for specific Chinese populations. But for the rest of us, I don't start many patients on carbamazepine. We usually kind of deal with that after the fact. As far as food,
I have a question for you. I've never understood, what does the herpes virus reactivation have to do with this whole thing?
So we don't fully know. So we think that it may either due to activation of the CD8 cells whenever they go in and they destroy or they lyse cells, that all of us, having previously been exposed to herpes viruses, have that in our genetic code, that then they may transcribe it and then progress more out into the open, and so that the immune system is activated more towards the drug, and so it enables the HHV6 to kind of propagate. Some of it may be that the specific proteins with HHV6 that are produced interact better with the drug, and we have some level of low production at baseline. We don't fully know. That's why there's an association, not a causation. We don't know why, you know, those specific infections versus, you know, EBV, CMV would be kind of more consistent. It would make sense, you know, we think about EBV with relation to penicillin having such a high rate of that kind of delayed nonspecific maculopapular rash, that it would also have a similar function, but we don't fully understand. The initial GWAS studies or genome-wide association studies for food allergy did show some HLA associations. They weren't super strong compared to, you know, like filaggrin with atopic dermatitis, but they did have an odds ratio that was 2.8, which so it was higher than the general population, and it typically is paternally inherited. So we can actually, you know, ameliorate the mother and say that it's all dad's fault. There was no correlation with TCR there. It's really isolated to the MHC side of antigen presentation, because they tend to bind some of the specific proteins a little bit better and kind of present it. There was also an association with milk and egg, and it may correlate with severe reactions, but the jury's still out as far as all of that goes. It's one of the important things to know that, you know, in this case, it is an association. We don't know if it's fully causation. So this is why some patients present with food allergy more often, or certain populations have it more often, or if it's kind of multifactorial as we all kind of think about it. In asthma, it may actually have more of a benefit whenever we're talking about endotyping, phenotyping our patients in the setting of occupational asthma or allergic asthma or AERD, because specific type 2 MHCs or MHC class 2s are more commonly seen in the specific disease. Right now, it's not commercially available outside of sequencing that you can actually get this information, but it may play a slightly higher role in the specific population. And then in atopic dermatitis, for a more persistent disease, a severe disease, a relapsing course, it was seen in 2 specific HLA genes. But once again, association is something that's important to know. We haven't fully kind of sussed out whether it's causation or anything else. Autoinflammatory or autoimmunity and infectious, the classic is ankylosing spondylitis with HLA-B27. And so for some of our rheumatologic colleagues, they probably know these way better than us. But rheumatoid arthritis does have a higher rate, and it kind of makes some sense if you think about MHC2s having either a better equipped ability to present self-antigen, but then also better able to bind CD4 or CD8 cells that then can activate them. It makes logical sense that they can progress onto either autoimmunity or higher susceptibility to infections. The ones that we probably have the most information on and you can commercially do relatively easy is your celiac disease, so your DQ2 and DQ8, as well as ankylosing spondylitis with HLA-B27. A number of other diseases, including Crohn's disease, ulcerative colitis, pretty much any autoimmune disease, including autoimmune thyroiditis, has been associated with specific MHC class 1 and class 2. But they do appear to be largely disease specific. And so that's one of the ways that we can kind of think about it. Not that it's, if it's positive, it doesn't, predetermine that they're going to go on to progress to have that specific disease, but it makes them at a higher risk. And then whenever it comes to HIV, chronic Hep B, leprosy, tuberculosis, and leishmaniasis internationally, there are some MHC classes that do appear to have higher preponderance of infections or kind of relapsing rates or susceptibilities. So how do we use this, right? So it's always important to think about what kind of situations that we see it clinically for, and then what we can actually use with the knowledge that we have. While it's great to kind of have a bar trick that we can bust out and throw out some knowledge, it's always important for us to be able to kind of think in light of the patients that we're going to see. So it's used the most in organ and hematopoietic stem cell transplantation. Currently, we match HLA ABC DQ-DR, so that's your 10 different loci. DQ is probably the least important of those, but we typically think about trying to have as many as a 10 out of 10 match, because that decreases your rates of graft versus host, as well as increases your rate for engraftment. So the more you are able to match those, it makes an unrelated donor not as good as a match related donor, but it's getting much better, especially now that we are doing high molecular identifications of these. Our colleagues over in oncology have probably been taking use of this knowledge the most. So this is not going to be about CAR Ts, which are slightly different, but they actually have a number of therapies in trials where for solid organ tumor, they take a Tcell, they have a specific TCR that is identified, directed at the neoplastic cell, and then they can either upregulate or downregulate the MHC with a fusion protein to activate T cells and NK cells to target cancer cells. So if they downregulate the MHC, then the NK cells will see it as missing self, and then they'll attack the cancer cells. And you can also use that as a way to activate T cells whenever you actually have a fusion protein that activates T cells. So if in the bottom here, they actually make a fusion protein that binds specifically to tumor cell antigens or tumor cell MHC, that then has a linker that will actually activate T cells. And so they can create these in vitro and then use them as a therapy in patients. And similar to CAR T, they do have a high rate of cytokine storm, where they actually will have mass amount of tumor lysis, but there are still tumor cells that can bypass a lot of this, a lot of these pathways, so that they kind of downregulate those antigens. So you actually have to have multiple steps along the way. So that's all I've got. These are my references. I know this is a lot. It was a very broad topic to try to get through. The word of the day is peptide. I'm happy to answer any questions. I don't know if I'm like the world's expert in this topic now, but I'm far from it. So I think that this has taught me how much we know, how much interaction we have to have with our immune system, how much built-in redundancy and effect there is. Happy to take any questions.
When do you get to the hard stuff?
Oh, that's next hour.
Next time?
Yeah.
Yeah. Well, my
I've
only
got to give you a
comment would
reason to
be,
bring me back. I've always got to give you a reason to bring me back.
yeah. I was impressed that you had 45 people listening to what essentially is a very difficult basic immunology that, for the most part, we can't use in the clinic every day. But
listening is a relative term.
Yeah. Whoever designed this system was drunk at the time to make it so amazingly complex.
It's
I
a
guess
wonder
one
that
question,
anything works
Clint,
well.
I know from how far back in phylogeny do we see similar things? When did the complexity of this immune system develop?
So going back, we've found MHC molecules, T-cell receptors, in pretty much every animal except for maybe single cell organisms. We do see them. So we found them in camels. We've found them specific for like infections like MERS in camels as a reservoir. We've found them in sharks. So we know that it has to split off. We found them in both amphibians as well as in mammals. So we know that it has to be kind of further up the chain. I'm not sure exactly. I just know that single cell organisms do not have them. So far enough, high enough up that it's relatively preserved throughout most species.
Let's see, someone typed in a question here. What upregulates MHC class 2?
So interferon gamma upregulates MHC class 2 in antigen presentation cells exclusively. So B cells upregulate MHC class. So they constitutively express MHC class 2, but then they can upregulate themselves anyways whenever a B cell receptor binds to a bound immunoglobulin that has an antigen. Sorry, not a bound immunoglobulin. Let me rephrase that. The B cell receptor, whenever an antigen cross-links and binds to B cell receptors, then it can upregulate its own MHC class 2. Antigen presentation cells are upregulated by interferon gamma.
Other questions? Nick is clapping.
I think that's either because he's happy that it's over, or he's like, oh man, this is way too complex. But so the reason that a lot of us didn't train with it or didn't know a lot about this is because we really only have had this information for the last 20 years, and it's been progressively growing. And it's specifically MHC or specific HLAs in disease have only been kind of more recent, within the last 10 or 15 years, kind of been discovered and published.
You can see clearly how you can use it for drug reactions that we have not understood. It's hard to see. I mean, we know about like the rheumatologic associations. It's helpful diagnostically, but you can't change your HLA type. So if you're born with that susceptibility, unless we figure out to genetically re-engineer people, that's just the way you are.
Yep.
Other comments before we sign off?
And it may also be important for a lot of the conditions if we're considering, so for systemic sclerosis, whenever they're transplanting patients, which is one of the new things that's coming out there, that hematopoietic stem cell transplant may be effective for systemic sclerosis, is not having a perfect match may be important, because if you, for a lot of those kind of regulatory diseases, if you transplant with the same MHC that puts them at high risk,
yeah,
you're not fixing the issue. And so you may not want a perfect match in some of the things, which then is counterintuitive in trying to make sure you don't have graft versus host disease, or you actually have graft engraftment. And so it's one of those like delicate new topics that our colleagues in transplantation are constantly thinking about. And I will defer to them for any other questions on that.
Yeah,
That's above
that's
me.
a very thoughtful comment. I hadn't thought about that. All right, Clint, thank you for tackling such a complex topic.
I'll be back next year to talk about quantum physics. I think that me and Drew can bounce that one off of each other.
Okay, well, as I was commenting, and Nick and others will remember when Drew's dad used to tackle topics of that kind. So you may pick up the mantle to do that.
Or we'll bring my dad out of retirement here.
Yeah. All right, we'll see if we can get him to wake up in the morning.
Yeah, be tough.
All right, so long, everybody.
You good?